right, so the biggest thing to remember is that if you're making a specimen and it's not transparent, you're not going to see anything. Right? So we're learning to make wet mounts with material that's a little bit more difficult than what we've done so far. We've done the pupil cells. So you're going to do those again, and then you're going to draw that. But I'm just going to show you how to make the other slides. Okay? So the bamboo is the only one that you're only going to draw under low power because the cells themselves are pretty thick and you can't get a cluster that's thin. So just do that under low power, otherwise you won't be able to really see anything. Okay, so you're not going to try to cut anything. You're going to take your razor, remember using the folded side is where you put your finger, and you're just going to scrape onto a clean slide. So you should be able to see that those little dusty things. Then you're going to take a drop of water and then you take your cover slip. Being sure to remove any excess water. The only ones that you're going to need to stain are your pupil cells, cheek cells, and the onion skin. So you can see this is laying flat. Now, if I were to make a big cut, I wanted to take a big slice. Mm-hmm. And I try to do the same thing. Look what happens to the slide, the cover slip. You can see how it's raised, right? So just like putting a microscope slide on upside down, you'll never be able to focus on it because it's not in the plane of view, plane of focus for that microscope. Okay? So that's your bamboo. The next one you'll have to do is your sponge. I mean, not your sponge, your onion. You can do these in any order, obviously. So with the onion, you don't even need the razor blade. That's why you may have done this in middle school. You just break the onion against the grain and then pull it apart. And you can see that the skin is translucent. You see that? And you're going to try to get it to lay flat and not fold over itself. If it's folded over itself, it has too many layers and it gets difficult because it'll be blurry because you're going through the layers with the microscope. And you just add a drop of water. It's often easier to try to get the air bubbles out before you put the cover slip on with the onion skin. Put your cover slip on. Now this actually is a little fat, a little wide. See where that air bubble is starting? That kind of indicates that this is a little thick for the microscope. It's lying pretty flat. So you have a couple options because you're going to stain this. You could kind of squish it down, or you can go back in and cut it. Remember, before you stain, you want to clean up all the water, otherwise it just gets really messy. So that's a lot better. No air bubble. And then when we stain, just put a drop. You don't want to touch the blue to the slide, and you definitely don't want to touch the water drop when you rinse it. So a minute, 40 seconds. You can help it along by putting the paper towel at the other side. It'll kind of draw the stain across the layer. I would let this sit a little, and when you view this under the microscope, you want to try to go to the edges because the edges are going to be the part that absorb this stain. Okay? And then make sure you rinse it so that there's no blue left. And you can see how if you touch the paper towel, it will get rid of the stain. And please don't forget when using the stain to make sure all the water is gone. That's the onion. And then finally you have your sponge. This isn't like the sponge that we use. This is natural sponge. So it looks like this. But if you put a piece of that down, you're not going to see anything. So you just want to take a few hairs and put them down. And have the water ready. You can even get it off with another tweezer. So you only want a few hairs. Even like that's too big, way too big. So you want to just, that's kind of big too. And then put a drop of water on it because they blow away. It just blows away. Put your cover slip on. All right, and finally you're going to do your cheek cells. So you'll take.
Do we each do our own cheek cells, or do we?
Only one person's going to do cheek cells, right? So when you do the cheek cells, whoever's doing the cheek cells, you take the sterile cotton swab, you roll it along the inside of your cheek like we did on Monday, and then all you have to do is touch it to the slide, put a drop of water, put a cover slip, then do the stain and rinse it. All right. So I want to make sure that everybody does at least one wet mount slide, and then you can do a prepared slide for the group of two. Each one of you does a prepared slide and then two wet mount slides. All right, so then you're going to hand in those sheets with the drawings on it, and then on Classroom there's a chart that you'll hand in, which is on the back. Was this alive? What was the shape? And is cytoplasm present? How do I know what cytoplasm looks like? If there's something floating in it, you have cytoplasm in the cell. If there's nothing in that cell, then there's no cytoplasm, right? Pretty easy. Okay. And remember, we talked about what the characteristics of what makes something alive. It has to have DNA. It has to have metabolism. Right? You might not be able to see the organelles, but if you see cytoplasm and floaty things, that indicates that it has the equipment to have a metabolism. And the prepared slides are blood and a paramecium. So the blood is human blood. Human red blood cells are not nucleated, but they are alive. They lose their nucleus to help them function. But all the slides have white blood cells in them, so those definitely have a nucleus, and they're a lot bigger, so it might be easier to draw those. Does everybody understand what we're doing? You're gonna have today and tomorrow to do this, and the test will be on Wednesday.